Hello researchers, we are the Gen Scientist group. Today, we're going to discuss the challenges in applying Mendel's laws in gene editing technology for better corn kernel size. Corn originated from a wild grass plant called teosinte from Mexico. Mexicans used selective breeding of traits like the kernel size, stem structure and more. There were some physical changes that happened over time caused by either the breeding or even environmental factors like temperature, stress and the amount of water or minerals available. However, selective breeding takes years for people to get the desired plant. Scientists then started wondering if there are gene modification methods that are easier, targeted and time-saving. They discovered a lot of modification methods, although most of them weren't safe, precise and accurate. Later, they found CRISPRs in archaea and bacteria. The scientist proposed that it serves as bacterial immune system that consists of repeating sequences of genetic code having a space between them. Gene modification of corn uses Mendel's laws as a foundation to guide how traits like kernel size are changed. Mendel's law of segregation helps ensure that the edited gene is passed to the offspring, while the law of independent assortment lets breeders edit multiple genes on different chromosomes to get the better kernel size. Well, although modification uses Mendel's laws as a foundation, kernel size is a polygenic trait which wasn't explored by him. This challenges his laws because not one allele but many alleles are combined to determine the kernel size.
So, what is CRISPR-Cas9? CRISPR stands for Cluttered Regular Interspaced Short Palindromic Repeats. This method is used by scientists to identify the DNA sequence leading to a problem and also be able to cut off or install new DNA sequences. According to Cost_Planted, 2020, this method is used to change a targeted base of a gene for mutations. It's precise because it has a targeted base of DNA. The method can also be used to edit several genes at the same time. This is important because it helps in editing multiple genes according to how they affect the kernel size, either reducing the amount or increasing. According to Zhang, H. A. al. 2021, and Michalovsky, 2022, the ZMark1, KRN2 and many more genes determine how big the kernels are. Food and Agriculture Organization, 2024, suggests that although gene modification tools like CRISPR-Cas9 can precisely alter genes, they risk off-target changes that can complicate how to predict. For example, changing the genes that help in increasing the coat of the kernel might also affect the structure of corn cob. You may be wondering what the effects of modifying corn, specifically the kernel size, are to the environment and society. According to DeVos and Swanenberg, 2017, research done on livestock reveals that there is no effect on the animal's health after examining different parameters like immune response, weight, body conditions and more, although there can be some physical changes without genetic variations. Referring to Walsh et al., 2011, research, pigs were given the BTMON 810 maize, insect-resistant maize, and others with non-GM maize for consumption in a 31-day period. Afterwards, they killed the pigs to test their intestines, kidneys and the amount of feed they ate. The results showed that the pig feed with GM maize consumed a lot compared to the one fed by non-GM maize. The pigs fed with GM maize had heavy kidneys, but no change was seen in the pig's blood biochemistry. They concluded by saying that there is no significant effect of the modified maize to the pigs genetically. Relating to the GM maize kernels, there might be no significant effect to the animals biologically because there was no evidence that the animals, mostly livestock, were being affected by GM foods. Although in consideration of the economy and satisfaction of the animals, the modification can be really important because farmers can feed a lot of animals using less maize due to the bigger kernel size, while they attain the same nutrients as the ones in the non-GM maize. According to Dynamic Earth Learning, 2020, people mostly benefit from the GM maize because they can be exported and brought to areas that don't have sufficient nutrients or land to grow them in to feed the people. They are also nutritious and healthy. Although these foods are really important, a lot of researchers believe that the foods aren't safe for everyone, but there is still lack of proven evidence. They propose that they might lead to certain allergic reactions, cancer or even resistance to certain antibiotics, which can lead to reduction of chances to heal from a bacterial disease. For example, in the US about 90% of the plants grown are from GM seeds. According to the Centers for Disease Control and Prevention, food allergies of below 18 children rose from 3.4 in the 1990s to 5.1 around 2010. Others also propose that the transfer of some proteins from one plant to another can lead to allergies. For example, if they transferred gene, that leads to a protein production, from a tomato to the modified corn seed for bigger kernels, then the person who had the allergy to the protein in tomatoes might also have the same allergy to the modified corn. Overall, Mendel's laws provide key principles for gene editing in corn, but modern editing must manage complex genetics and environmental impacts. Responsibility. Groman et al. 2021. Editing corn genes can improve food supply and crop resistance but needs careful study of effects on animals eating the corn and soil ecosystems to avoid harm. For humans, gene-edited corn can enhance nutrition but requires safety check to prevent allergies or any other risks like other diseases. More study is still needed to know whether the underground organisms are also affected because most studies focused on the above-ground creatures. According to Noack et al. 2024, there are still some significant gaps in our knowledge on the direct effects of GM plants on non-target organisms like bees, flies, snails, aquatic organisms and many more. Modify, honor, make the world a better place.